I feel as though, partly through white male privilege, and partly by virtue of being an independent artist, most of the decision-making in my life has been done through intuition. In retrospect, one often finds patterns. It is with this attitude that I invite the reader to take the long view and cast their mind's eye like a net across time, to see as others have seen before you, no matter how repulsive that may be, and draw from the great annals of human history set aside that which will be crucial for the next age. Indeed, the habit of setting aside may well have been one of the earliest activities of our species. Gathering and storing nutritious foodstuffs against the coming hardship. The late science fiction writer Ursula K. Le Guin drew our attention to the work of women's studies professor and writer Elizabeth Fisher, who reasoned that woven baskets or other containers must have accompanied this primordial habit of gathering. In much of her work, Le Guin encourages the reader to address what gathering might mean. Which concepts, kin and cookery are you, the reader, about? What has utility in the now? What can be shelved for another moment? This sorting process of life extends from the DNA in our cells to the materials we gather around us, and to the ideas we attach to reproduction and variation. In the carrier bag theory of fiction, 1986, Le Guin evokes the jug or basket, bottle or leather bag as a protective apparatus for the storage of perishable matter. Fiction, in fact all language, is no exception to this task. Words can be baggy and shaggy, letting meaning slip out all over the place. Or else they can be tight and stringent, with barely any room left to allow for meaning to be topped up or added to over time. This book is a work charged with the goal of exploring cultural evolution, and it would be remiss of me not to improve on my previous efforts. Queer, fag, witch. These are examples of stringent language that, no matter how frequently they are subverted, retain their violence. One could list many more in many different languages. So the assessment of which forms of language make for beneficial containers should also be burdened with the problem of which kinds of knowledge serve whom. What Le Guin was at pains to demonstrate in works such as The Dispossessed, 1974, was the embeddedness of human life within the material world and within the worlds of others. If we take foraging, a process of selection, as part of the task of preparing for material and spiritual hardship, the contingency of one life form or concept on another should also remain at the forefront of this approach. In Darwinian evolutionary terms, Elizabeth Fisher and then Ursula Le Guin's concept of gathering would be akin to genetic variation, a nexus of experimental forms from which an organism, brackets here the writer, close brackets, selects, grows and eventually evolves. One of my tasks then is to prove, once again, that biopolitical and cultural forces have always intersected with the materials to shape human destiny. But as critical thinker and philosopher Donna Haraway argues, that the biosphere encroaches upon us is increasingly hard to ignore. So, while I am interested in unpacking certain terms, such as gathering, variation, selection, adaptation, and evolution, in cultural and political registers, as I have been working through the particular problems of this project, I can't help wondering, what else is there to be done? With whom? Emerging from an entire year of writing in lockdown, and isolation within an isolation, I have to ask, what just happened? The unprecedented contemporary restrictions on civil freedoms oblige me to think deeply. What else has all our theorizing and protesting and postulating at conferences been for, if not to develop critical thought? Or have we all become too comfortable, too successful, too bored? If I think in the political, restrictions upon the act of gathering as bodies at a public protest reveal the state of my political subjugation, and which possibilities exist outside of the state's monopoly on violence. Gathering is also central to how state and non-state entities collate and compare data points, and it is these two issues of publicness and big data that combine to demand a serious discussion around how each are regulated in the modern world. These are difficult subjects to address in the heart of the worst days of the pandemic. However, I hope that the political, historical, and conceptual content of this book prove to be critical and careful enough as to contribute to the discussion around what happens when a public health disaster becomes a civil liberties wasteland, and the danger of permanent restriction that this situation provokes. As the mutations of the virus pile up, each progressively more successful variant travels faster and infects our host cells more efficiently. Variation and selection have always been around us. Perhaps the most controversial term within Charles Darwin's evolutionary biology is that of selection. Selection has been tied to the concept of survival of the fittest. Selection implies some form of reason or intellect. Where this argument becomes particularly problematic, it also alludes to the concept of quote-unquote intelligent design. Selection has therefore been used to invoke the workings of a godlike and divine consciousness, a paradoxical reason above reason, in German Wahnsinn, a mad sense. Culturally speaking, selection, like variation and adaptation, are easily integrated into a view of cultural reproduction as an ecology of ideas. Each of these linguistic registers, cultural, political, and biological, will be consulted below. To gather different knowledges is appropriate both for an era of great change and to the practice of philosophy itself. Gathering, sorting, and comparing was described by the late and brilliant anthropologist and economist David Graeber as simply what humans do. For any philosophy worth its salt, this entails the taking of positions, choosing sides in the knowledge that maintaining them often requires drawing on all kinds of experiences and arguments other than one's own. Those who are able to risk having their own truths taken to task by opposing beliefs know that it is often not so much the core tenets of an argument, but the way in which it is made that reveal the strengths and weaknesses of our positions. To ignore the historic reality that wealthy Jewish court financiers could not become part of the proletariat because of their wealth, and were not accepted by the bourgeoisie because they were Jewish, is anti-Semitic. To me, this is one of the fundamental revelations of Hannah Arendt's Origins of Totalitarianism, 1951, which I will elaborate upon in detail in Chapter 5. For now, it is worth bearing in mind that the dynamics of wealth and power in the transition from feudal courts to emergent European nation-states relied upon privileging a few court Jews while isolating the vast majority of European Jewry. This meant that centuries prior to the political anti-Semitism which gained traction in Europe in the late 19th century, Jewish persons were stigmatized as both impoverished and members of the elite. Indeed, the so-called emancipation of Jewish peoples under Napoleon in the early 1800s was never complete, and in many instances served to isolate Jewish communities even further. As Arendt writes, quote, "The fundamental contradictions between a political body based on equality before the law and a society based on the inequality of the class system prevented the development of functioning of republics, as well as the birth of a new political hierarchy. There is no doubt that the nation-state's interest in preserving the Jews as a special group prevented their assimilation into class society." Hannah Arendt, The Origins of Totalitarianism, 1951. One could use these historical facts, however, to depict a completely different version of reality. But it would always be wrong, for it is no longer sufficient simply to state that these are my truths, now tell me yours. There is nothing inherently wrong with the fact that gathering brings us together as actors or agents where selection sets us apart. So long as I do not allow fundamentalists to prevent further gatherings, new combinations, new contestations around which aspects I choose to incorporate into my being. Selection is a part of life. One cannot be for everything. The cultural and scientific study of evolution is, if you like, just another way of approaching the biological process of gathering and selection, and vice versa. Moving between the biological and the historical is a means of operating at different scales. This allows me to focus and refocus my attention on my subjects, mutation and epigenetics. Like Rick Deckard in the cinematic adaptation of Philip K. Dick's Do Androids Dream of Electric Sheep in 1968, Blade Runner in 1982, zooming in and then tracing back to the larger picture, revealing the next step of the plot. Chapter 2. Introducing Thale Cress and the bamboo shark. Okay, so here we've got the first illustration of the publication. On the left side of a two-spread black background, we find the scientific representation of a thread of DNA and its magnification first into the scale of a gene, which is a bracketed section that's composed of different codings of DNA, let's say. And then it's magnified again to see much more closely how these individual DNA patterns are composed. So they're composed by the chemical base pairs adenine, thymine, guanine, and cytosine. To the bottom of the illustration, you start to see how this is kind of these genes are wound up to make up a thread of DNA, and one can see that at this scale there is a chemical tag sitting on the histone tail, which is a little kind of tail that comes off of the DNA section, and that's where the epigenome is present. And so that determines through if it's activated or silenced by environmental factors, all kinds of different factors. That's where that occurs. That's going to affect cell replication, but that isn't in this particular diagram. This diagram then shows how these histones are wound up to become an individual chromosome, which is probably the most kind of familiar sort of X shape that you would imagine when you think about these kinds of diagrams. On the following page, page 24, we have a depiction of a cell replicating, again in a very diagrammatic form. And so at the bottom of that image, you have the cell recomposing. And at the top of that image, you have it opening up and splitting into two. While I've been living here in the Czech countryside, about an hour outside of Prague, in a small village called Želiv, I've been punctured by a number of insects. Mostly by mosquitoes, but possibly also horseflies, though not yet, thankfully, by ticks. Either it's horseflies or else some of the mosquitoes are carrying something that my body has a strong reaction to, making the site of punctuation swell up into a broad lump. There have been bites that have made it uncomfortable to walk. Such is the swelling. It makes me think about the relationship between my environment and my inner world. When I swat a mosquito, a small cartoon mosquito pops up in my imagination, pleading me that, as a mother, the mosquito is only trying to draw a few drops of blood to feed her children. I don't know why it's a female mosquito, but I guess the struggle of a single mother adds to the pathos. I swat them away, as I'm not convinced of being the sole link in a food web that keeps the mosquito family alive. Followed by the next level up the food chain, a frog maybe, and so on, until the final victim of my needless swatting is a mammal larger than myself. But I do stop to think about it. I spend a lot of time just thinking. Sometimes too much, I'd have to admit. Overthinking things can arrest one's development. Before attempting to answer some of the important questions raised by a comparative exercise of thinking social and philosophical problems through the science of biological mutation and epigenetic expression, I would like to share some of the terms and mechanisms here at play. I have elected to explore the science first, and critique the fraudulent racist ideas attached to biological arguments around cultural predetermination thereafter. Of the different types of mutation that occur, phenotypic mutations are perhaps the easiest to demonstrate, model and describe. This is because there are, by definition, alterations to the visible physical properties or function of an organism, such as leaf or hair pigmentation, or an organism's growth and stature. How does this work? Well, the development and function of an organism is largely controlled by its genes. Genes are selected sections of an organism's full DNA sequence, known as the genome. All cells reproduce by making copies of themselves. They self-replicate in an ongoing process that requires sustenance, energy, and nutrition. During this process, a cell divides, creating two versions of itself, making a copy of the genetic material within that cell. Sometimes the process of replication incurs errors or deviations in the transcription of genetic code, and these are known as mutations. Mutations can lead to changes in the structure of an encoded protein, or to the complete loss of a given gene during replication. Mutations can occur in a number of ways. To recap, while cell replication aims to create an exact replica of the cell, mutations of DNA during replication mean that the copy will contain errors in its DNA, brackets, and therefore differences in its proteins, close bracket. This will then affect the character of the cell itself. As mentioned above, an organism's genotype is the complete set of genes that make up, brackets, literally create, the individual organism. Whereas the phenotype are the observable characteristics and physical appearances of that organism. This means that while we can talk about a cause and effect relationship between genotype and phenotype, the former made manifest within the appearance of the latter. But changes in cells as they replicate can also be caused by external factors, as well as changes to the programming within an organism's cell itself. There are then a number of different ways in which a cell can create versions of itself with novel characteristics. I'm going to focus on two particular approaches, or modes of development. And although the outcomes they produce may at first appear to have a lot of overlap, they are distinct from one another in important ways. Two significant modes of change occur when a cell replicates. One involves epigenetics, and the other, mutation. Epigenetic changes can be brought on by a series of chemical tags which sit on top of the epi, a chain of wound-up DNA called histones, which determine the way the genes are read. More on this below. The other form of developmental change I want to draw upon is mutation. Strictly speaking, mutation only occurs at the level of the genome. Mutation in organic life, plant or human, is an alteration to the hardware, the DNA of the organism. Whereas epigenetically triggered development is equivalent to the software running in a replicating organism. When a cell divides and replicates, The new cells are called daughter cells. Fidelity to or variation away from the mother cell is determined by which genes are turned on or off, expressed in the manifestation of a new daughter cell. Different filterings of the genetic code, whether from software, epigenetic programming, or hardware mutations to the code, emerge as expressions. There are, overall, many ways in which genetic variation occurs. Mutations happen when the manner of gene translation that takes place as the cell replicates produces copies with novel interpretations. As mentioned above, this can be the product of an environment that triggers either overcompensation or the silencing of some genes within that cell. One of the key differences between mutations, triggered either by an environment or by random internal process, and epigenetic variation, is that mutations represent, in a sense, the deviant exploration of the organism. Epigenetic tagging, the epigenome, on the other hand, is a form of cell memory. The epigenome is a function of the body that instructs various cells to reproduce the materials needed to create, for example, a hand or a leaf at any given time. So when a plant loses a branch or lateral shoot, it will draw on its epigenome that remembers how to read the plant's DNA in order to sequentially reproduce its lateral shoot, leaf, and eventually the plant's flower and seed. Footnote 1. Plants are particularly dependent on epigenetic mechanisms to express diverse phenotypic responses from the same genome sequence. Plant epigenomics, deciphering the mechanisms of epigenetic inheritance and plasticity in plants. Genome Biology, 2017.